So we've talked about mitosis. Next week, Wednesday, just to remind you, we are taking a test on mitosis, meiosis, and the cell cycle. So major steps of meiosis we very quickly went through yesterday in the notes because we are coming back to reinforce, those are pens I should return. We're coming back to reinforce these today, so here it is. So when we talk about mitosis, we start with one diploid cell and it produces two genetically identical diploid cells. There was an activity that we've done in the past, and I'll show it to you because I think it does a nice job of modeling things. So we used to do an activity, last year we didn't do it because of COVID, this year we kind of didn't do it because of COVID and time, but there is, we did, that's weird, it says it is airplane. So we modeled mitosis using a form of food, and we used gummy worms. So the idea is we went and we started with 4 gummy worms, and the gummy worms have colors, so we call those genes. And we got this gummy worm from mom, we got this one from dad, and they had the same genes, hair color genes. Mom gave the red hair allele, where dad gave the orange hair allele. And then we had the eye color allele, or gene, with 2 different alleles. We had the toe color, obviously, toe color allele, and then the finger allele. So these are the chromosomes that we get from mom and dad. And then we went through and drew in the different phases. Now this is interphase, and this is prior to S phase, so prior to replication. But what's wrong with our model? If our gummy worms represent DNA, what form should our DNA be in during interphase prior to S phase? Chromatin, spaghetti, right? It doesn't look like spaghetti. Okay, but there's a kind of limitation of our model. But what happens during S phase? DNA doubles or replicates. There's my chromosomes, right? And this is what happens during S phase is we get the other half. We get the sister chromatin. We have some centromeres, okay? And they are genetically identical to one another. Prophase, the nucleus dissolves. Metaphase, the chromosomes go to the middle. Anaphase, the chromosomes pull apart. Telophase, cytokinesis, to end up with 4 gummy worms in each cell, and we have 2 cells. Now we started with 4 gummy worms in a cell, and we end up with 4 gummy worms in the top and 4 gummy worms in the bottom. And these are daughter cells, and this is the parent cell. What's true about my parent cell and my 2 daughter cells? They are genetically identical. Boy, those sure look the exact same, right? And that was the idea and the intent of this. So mitosis is when we take one diploid cell and we produce 2 genetically identical diploid cells. But today we're not talking about mitosis. We're going to talk about some of the similarities between mitosis and meiosis. Today we're talking about meiosis. And meiosis occurs in is a way to produce sex cells. Okay? Meiosis produces sex cells. Mitosis happens in your toses. Mitosis, toses. I don't know. There was something with some toes. I was trying to remember that mitosis happens in your toes, which is a part of your body, but I don't think that was quite right. Okay? Mitosis happens in all of your body cells, with the exception of sex cells. Meiosis happens in sex cells and only in sex cells. Okay? So when we talk about meiosis, there is definitely some similarities, but the big thing is we start with one diploid. We start with one diploid cell. That's what we start out with. Okay? Very big picture, we start with one diploid cell. And our diploid cell in interphase looks very similar to the diploid cell that we drew in interphase of mitosis, and that's because the same stuff happens in interphase. Okay, so we have a nucleus, and we have that bowl of spaghetti, which is chromatin. Okay. So what happens in G1? Cell grows. So the cell increases in size in G1. And S phase allows the cell to duplicate the DNA, and then in G2 we're going to say they duplicate the organelles. We talked about how some texts have that kind of differently, but we're going to go with it. Okay, red and green. That's the diploid cell. Trust me, it is. Okay, then prophase. Now there's meiosis 1 and there's meiosis 2. Meiosis 1, meiosis 2. Very similar. Some key differences. So, meiosis 1 happens first, just for the record. Meiosis 1 comes before meiosis 2. But meiosis 2 is dealing more with homologous chromosomes. So as long as you kind of keep thinking homologous chromosomes, homologous chromosomes, homologous chromosomes, for meiosis 1, that's going to be helpful. So let's talk about some big things. During prophase 1, each chromosome pairs up with its corresponding homologous chromosome, and it forms this thing called a tetrad. Okay, it forms something called a tetrad. I like to think as this process being a buddy check. You guys ever go to summer camp and you had buddy checks at the swimming area? You guys know what I'm talking about with buddy checks? Okay, then let me explain what my summer camp experiences were. You go swimming, we want to make sure people don't drop. And trying to keep track of a bunch of little kids is challenging for lifeguards. So you get a buddy, and every now and then they randomly call out, buddy check! And then you gotta find your buddy, because you're always supposed to be kind of close to your buddy. But you gotta find your buddy, and you go over and you grab their hand, and you hold your buddy's hand. And then, like, someone goes, oh, we should have 15 buddies. And you go, and as long as there's 15 buddies, 15 pairs, we got all 30 people. If I'm sitting here going like this, and they'd be like, hey, where's your buddy? And you'd be like, I don't know. Hopefully your buddy left and is back at the cabin or campsite or something. Hopefully. If not, then we got issues, right? And then lifeguards have to do some searching. But hopefully, the idea is buddy check, grab your buddy, and buddy, you get close to your buddy and buddy check. Hey, I got my buddy, we're here. That's what I think of when I think of homologous chromosomes. Ever play the game Tetris? I used to play Tetris on Nintendo or on a Game Boy, which is really, really old, and I'm dating myself. But how many squares or boxes does every single piece in Tetris have? It has 4, right? So when we talk about tetrad, it's 4. And what we have is we have 4 chromatids. Okay? So we're going to draw our cell just like in prophase of mitosis, the nucleus dissolves. If you want, we can label it, or we can. I think right now the dash line is we're probably pretty good without labeling it. And I'm going to draw a total of 4 chromosomes. And I'm going to show you kind of how I draw these. So I'm going to draw a little green chromosome down here. And I'm going to draw its buddy, a little red chromosome, next to it. Then I'm going to draw a big red chromosome up here. But I'm going to do something a little bit different with my chromosomes. So kind of watch this quick. Okay, so I'm going to make one of my reds a little bit shorter, and that's because go like this, and go like this. Okay? So I did something like this. Again, I have a buddy check, but with my big chromosomes, there's this thing called crossing over. So what I had is a big green sister chromatid, but then what happens is because they overlap, they're going to do a swap. It's called crossing over. And my green actually goes this way, and my red chromatid goes like this, and then it kind of bends, because these are actually going to flip. Okay, I'm trying to show a crossover event. Okay, so right here is called crossing over. So at this point, portions of the chromosome swap, and this causes, it's called crossing over, which allows for genetic variation. So we talked about genetic variation within organisms and genetic variations within populations. Sexual reproduction increases genetic variations within a population. The combination of two different parents' genetic information causes their offspring, you guys, to be different genetically than mom and dad were. Okay, that's sexual reproduction increases genetic variation. But within the production of sex cells, there's genetic variation occurs too. So that means the sex cells that you make are not identical to each other. There's genetic variations within the sex cells that you produce within your lifetime. And one of the reasons, or one of the ways we get genetic variation within sex cells is this crossing over. So what the idea is this is a chromatid right here, and what this chromatid was all green is now got a little bit of red on it, and this red chromatid that was all red now has a little bit of green on it. We do a swap. And we'll kind of see that a little bit in the next step. Okay, so we get genetic variation there. Then we go into, I should say that, we go to the next phase, which is metaphase. And what was the other M word that goes with meta? Middle. And we're still going to have things go to the middle. Just for the record, in this one, my middle is going to be like Grand Meridian, or the international date line, north-south. I'm going to draw them in the middle this way, and then they're going to pinch. They're going to pull this way. Okay, that's my plan. So we have things moving around, and we get our chromosomes that go to the middle. Meta, middle, meta, middle, meta, middle. The difference is now the homologous chromosomes. So I'm going to draw my big red X over here. And then I have a little bit of a green on, oops, don't do that yet. It's okay if you did, but don't do that. I'm going to flip my red. Okay, so I'm going to have a little bit of green, and then my big red there. This is supposed to be all red. That's all red. Obviously, that's all red. Okay, but I do have a little bit of green right here because a crossing over event happened, and then I have a little bit of red. Okay, so I have my big green and red chromosomes in the middle, and again I'm going to draw them in the middle this way. They line up next to each other in the middle. Now, how do these line up? Does this go green, red, or red, green? It's random. It's a flip of a coin. If I flip heads, green goes left. If I flip tails, red goes left. I'm making that up, obviously, but the idea is that flip is random. And I flip a coin for the big chromosomes, and then I flip the coin again, and I flip heads, which I don't remember what I said, so you're going to have to tell me, and you can make it up if you don't remember. Heads said that which color went to the left? Red or green?
Red.
Red! Okay, so because of that, my little red chromosome goes to the left, and then that means my little green is going to go right. Did my first coin flip have any effect on my second coin flip? Or were they independent events? Independent events. Okay, good. They're independent. Okay, now why did they go to the middle? There were those ropes, and what were those ropes called? Yeah, so we have these spindle fibers. spindle fibers. The spindle fibers attach to the chromosomes and they line up individually and randomly. And then this should say in, not on, in the middle of the cell. I guess middle. Middle is probably fine, right? In the middle. So the homologous chromosomes line up in the middle. Here's my middle. Anna apart, Anna apart, Anna apart. And I'm drawing myself, oops, I'm sorry. This is called independent assortment. And allows for further genetic variation. Okay, so let's talk a little bit about that. So I have 4 chromosomes. This could go, ignore these crossed over things. This could go green, red, green, red. Green, red, green, red. It could go green, red, red, green. It could go red, green, red, green. It could go red, green, green, red. I think I have all the possibilities. There's 4 different combinations how I could arrange these. Okay? So that means with 4 chromosomes, I have 2 pairs that can arrange differently. I have 2 to the 4th power of how many different variations I can get just by flipping the coin and how these mom versus dad homologous chromosomes line up. We don't have 4 chromosomes in our cells. We have 46 chromosomes, which is 23 pairs. So every time these homologous chromosomes line up, we independently randomly flip a coin, and that determines whether it goes green, red, or red, green. So based upon that alone, we take our red or green, 2 possibilities, raised to the 23rd power. There's 8,388,608 different genetic combinations of those 23 pairs of chromosomes of red, green, green, red. So that means in your body, when you make sex cells, independent assortment alone can create 8,388,608 genetic variations of sex cells. So what that's saying is, if you were to look at 2 sex cells under a microscope and you were able to look at every single chromosome, which would be not doable, but whatever, go with it, the likelihood of having 2 of the genetically identical ones is very, very small from that standpoint. Okay. Now that's in you. Okay. Then if you're going to have a child with another person, they also have that many different genetic combinations of their chromosomes. So the likelihood of you and your significant other having a child, 2 children that are genetically identical by independent assortment alone is not very high. It's possible, but not probable, we would say, right? I think you add those 2 numbers together, and it'd be like 1 out of 16.8 million probability that 2 sperm and eggs produced and fertilized would create 2 children genetically the same. Not very likely. That's independent assortment alone. That doesn't account for crossing over. Because now it's not only this is different than this, it's these 2 are going to produce different ones as well. Okay, so that's that idea. It increases the genetic variation of these sex cells that can be produced. Okay, now we get into Anna. Anna apart, Anna apart, Anna apart, just like meta was middle. Anna apart. Okay, and like I said, when you're thinking about meiosis, think about homologous chromosomes. So homologous chromosomes go to the middle. And Anna apart, what pulls apart? The homologous chromosomes pull apart. So I start to elongate that cell a little bit. And what I get is I get a big green chromosome with a little bit of red to the left, with a little red chromosome to the left. Then to the right, I get a big red chromosome that has a little bit of green on the bottom. And then I have a little green down here. And what's pulling these spindle-fibers? Good, spindle-fibers are pulling these apart. Okay, but we get these homologous chromosomes pulled apart. So spindle-fibers pull the homologous chromosomes towards the opposite ends of the cell. And apart. Telophase, I'm going to slash it with cytokinesis, like we did before. So what's going to happen is we start to get a pinching of the cell membrane. That's the cytokinesis part. And we get kind of a bubbling starting to form. The nucleus starts to reform. To be honest, I don't know why, because it's going to dissolve again right away. But the nucleus starts to reform. And what we end up with is we end up with a cell that looks something like this. Now all I did is I divided that. Now, kind of imagine for a second that it actually pinches completely and the nucleus completely reforms. And we end up with how many cells? 2. There's 2. Now, how many chromosomes did we have up here in a cell? We had 4. And down here we have how many in each cell? And 2 is blank of 4. Half of 4. So if we started with a diploid cell and that was 4, but now we have half the number, what do we end up with? And how many of them? 2 haploid cells. It separates into 2 haploid cells. We start with the diploid. We end up with 2 haploid cells that have half the genetic information that we started with. Meiosis 1, homologous chromosome cell. Homologous chromosomes match up. Homologous chromosomes go to the middle. Homologous chromosomes pull apart. Meiosis 2 is pretty much identical to mitosis. It's almost identical to mitosis. So for prophase 2, remember, these cells will now have divided, so I now have 2 separate cells. The nucleus was reforming, so I should have a nucleus. However, that nucleus immediately starts to dissolve again, so don't ask me why. It reforms. But it's going to go through, and we're going to show them the nucleus dissolving again. And the cell immediately goes into prophase 2. Prophase 2. Without replicating the DNA. So we are not going to replicate the DNA. So all I'm going to do is I'm going to draw this chromosome combination here and this chromosome combination here in its separate cell with the nucleus reforming. Okay. And this has the same events as prophase from mitosis. So the nucleus dissolves, spindle fibers start to form. It's already condensed. Meta, middle, meta, middle, meta, middle. I now have 2 cells that are going through metaphase. This time I'm going to make my north and south poles be where the spindle fibers attach. So I'm going to line up my chromosomes along the equator. So I am going to draw my...
We can't see that.
Oh, thank you. I am going to draw my big green chromosome along the equator and my little red chromosome along the equator. Then I'm going to draw my big red chromosome along the equator and my little green chromosome along the equator. So what happens? The chromosomes, oops, that is not how you spell chromosome. Chromosomes. The chromosomes line up in a similar way to the I'm going to scroll out the similar to metaphase of mitosis. The chromosomes line up in a similar way to metaphase of mitosis. So now they're lining up in the middle, but now this is the middle. First middle we drew is like this, the second middle is like this. And it's really for spacing purposes on our bubbles. Meta middle. Ana apart. In metaphase of mitosis, what pulled apart? In mitosis and metaphase, I'm sorry, anaphase of mitosis, what pulled apart? Anaphase, what pulled apart? The sister chromatids pulled apart in anaphase of mitosis, and that's exactly what happens here. Now be mindful of my X, right? Because this is going to go north and south. So this chromosome, the chromatid on the northern part is solid green. The way my picture is drawn is solid green. But then on the southern part, it's mostly green with a little bit of red. My little chromosome, my little red chromosome, also pulls apart. My big red chromosome with a little bit of green, the chromatid that has a little bit of green on it, that was in the northern part, went north. And then this bigger part went south. Little green, little green. The sister chromatids separate and move cells to the opposite ends. Why? Spindle fibers. Okay, so I'm going to slash telophase and cytokinesis, and I'm going to scribble out cytokinesis down here just for the record. Okay, sister chromatids pull apart. Then in telophase, you start to pinch, nucleus starts to reform, and the DNA is going to start to uncoil into chromatin form. I'm going to kind of skip the chromatin part, just because it helps kind of illustrate a point that's an important point. So this is going to start to pinch inwards right here. This one's going to start to pinch inwards right here. So we kind of get something that looks like this. Oh, I got a really funny joke I should tell you right now. What did the 0 say to the 8? Nice belt. Get it? It's just not funny enough to laugh. It's okay. That makes me think of that though, right? Taking a belt and this was a 0, now it's becoming an 8. It's almost as good as what did one snowman say to the other snowman? Do you smell carrots? Okay, moving on. moving on. All right, so, sorry. I think it's funny that no one really laughed at my joke. Actually, kind of a laugh of pity out of myself, but that's okay. I didn't come up with it. I just told it because they were funny. I thought they were funny. Typical dad joke, you know. I am a dad. So you get a nucleus starting to reform. You get our nucleus starting to reform. And it has the same events as telophase from mitosis. And what we end up with is, now I'm just going to draw this chromatid in a straight line. I got a big green, and I get a little red, and the placement of those is random and doesn't really matter. And I get a little red in the bottom one, and I get a big green one that has a little bit of red on the bottom because of that crossing over event. Then in the top one over here, I get a big red with a little bit of green and a little green. And I get a little green and a big red. Now the reason I scribbled out cytokinesis down here is because I like to draw my final pictures. I end up with, now these all should be similar in size, but I kind of didn't do a great job there. The nucleus is now reformed. So I'm going to draw, I'll also label that really quick. That's the nucleus. Okay. And what really happens is we should probably really be drawing this in chromatin form, but I'm not, because it helps illustrate a point that is an important point that I want to make. So I'm just replicating how I drew these chromatids up top. Okay, so I end up with 4 cells. I started with 1 cell, and that had 4 chromosomes. I now have 4 cells that have 2 chromatids in. So, if we started with 4, and now we have 2, these are called what kind of cells? They're daughter cells, true, but they have half the amount of genetic information, therefore we call them haploids. And how many haploid cells do we have? 4. Now let's take a quick look. Big green, little red, little red. Are these 2 cells genetically identical to one another? Why not? There's a little bit of red right there, right? There's that crossing over event that happened, which these 2 sex cells, these are sex cells. Call them sperm, call them eggs, I don't care. These 2 sex cells are genetically different from one another. And are these 2 the same or different from each other? And what about these 2? 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 They're all different, right? So the idea of meiosis 2 results in 4 haploid daughter cells that are genetically different from one another. I end up with 4 genetically, and this is just restating the book, so you don't have to write it if you don't want to. 4 genetically different haploid sex cells. 4 genetically different haploid sex cells. So in adult, well not adult, in males, human males, that are producing sex cells, sperm cells, 1 diploid cell will produce 4 genetically different haploid cells, sex cells, which are sperm cells. In female humans, it does produce 4 genetically different haploid cells. But because of the division, it actually, of those 4 haploid cells, only one of those cells is actually what we call a viable egg, which is capable of fertilization. So of those 4 cells, only one of those eggs, or one of those haploid cells, are capable of fertilization. The length that these processes occur vary. Okay, the production of sex cells starts in humans around puberty, right? That's kind of the onset of this. And for males, it pretty typically happens till the day they die. They're producing sperm cells. And for females, it happens till menopause. And menopause is kind of the stop of the production of eggs. Okay? So that's the production of sex cells. And we produce these sex cells that create genetic variations. And then what happens to those sex cells is they may combine to fertilize and make a new offspring. Okay? So, just like I did it with gummy worms with mitosis, I also did it with meiosis. So again, this is just kind of visual, it's kind of nice to look at. Same thing, hair color, eye color, stuff like that. And everything starts the same. Again, this is really in chromatin form, our DNA hasn't replicated. Okay, now it's replicated. Now once we get to prophase 1 is where, buddy check, we form a tetrad. Those two homologous chromosomes line up, these homologous chromosomes line up. But what happened to this gummy worm and this gummy worm is they got chopped and they did a swap. Crossing over event occurred. Same thing happened to these two. Instead of being red-blue, it's now red, yellow, green, blue, where it should have been green, yellow. They swapped. Okay, they did a crossing over event. Then, prophase 1, meta middle, the homologous chromosomes go to the middle, ana apart, the homologous chromosomes pull apart, telophase, cytokinesis, and we end up with two cells that have half the number of chromosomes. We started with 4, we end up with 2, we end up with 2 haploid cells. And then we go prophase 2. Prophase 2, metaphase 2, they line up in the middle, ana apart, anaphase, sister chromatids pull apart, cells, and we end up with 4 genetically different cells. Sex cells, haploid sex cells, egg or sperm. Now, viewer discretion is advised. I'm going to show you a slide of 2 worms going through fertilization. Well, not worms, 2 cells going through fertilization. There it is. For mature audiences only. This is called sexual reproduction when a sperm cell and an egg cell combine. So we produced a haploid cell, we produced a haploid cell. 2 plus 2 gives us a diploid number of 4. We restored the haploid number. This is a fertilized egg. What happens to that fertilized egg now? What process occurs after that? You were once a single egg, fertilized egg, but now you're trillions of cells because of mitosis. That happened again and again and again. Okay, so that's all things normal. Sometimes things don't go right during the process of meiosis. So if we look at, and we say, oh look, this is the same, different gummy worms, but the process is the same. Oh look, there's a crossing over event. Oh look, there's metaphase 1, buddy check. The homologous chromosomes line up in the middle, but uh-oh, anaphase 1. What happened here? What doesn't look right in this picture? Anaphase 1. Yeah, they didn't divide properly, right? These should have gone up and these should have gone down. We call this non-disjunction. So what happens when we go through the results, everything else kind of progresses as normal, with the exception of when we're done. The top 2 cells have less chromosomes than they should have, and the bottom 2 chromosomes, or the bottom 2 cells, have an extra set of chromosomes that they shouldn't have. If this sperm cell or egg cell fertilizes with another sex cell, or this sex cell fertilizes with another sex cell, there could be some potential issues. This one is going to have less chromosomes than it should. We call it a monosami instead of 2 of a chromosome. We have monosami. Instead of having 2 after fertilization, we get a trisomy. Okay? So the idea is if we were to go through, we would get non-disjunction. We get an extra set of chromosomes, which an example of a trisomy is Down syndrome, is a cause of an extra chromosome. Please put your colored pencils away. Have a marvelous weekend, everyone. We will see you